A very good morning to all of you. I welcome you all back to my channel, Studika Enzyme. How are you all? I hope you would be learning better. Yes, so here we are beginning with the next part of this chapter, Principles of Inheritance from Biology Grade 12. And we will be continuing this part 3 after we have done monohybrid, dihybrid cross, non-Mendelian inheritance. Now we will be moving ahead towards linkage, recombination. We will see all these phenomena here. Let's proceed. Okay, so now you have heard that the father of genetics is Gregor George Mendel, who stated that the traits or the characters or the physical features that we see are because of the genes or the factors. But he was little proved by that time. And then onwards in 1900, three scientists, Hugo de Vries, Correns and Schenmark, they worked independently and rediscovered that the Mendel, whatever mathematical work that Mendel had done, whatever mathematical conclusions Mendel had drawn, whatever laws of inheritance that Mendel had given were all correct. So they stated that the factors that he had been telling about were actually the genes which are present on the chromosomes. So they verified the very same thing. They verified that whatever genes were available, they are factors, right? That Mendel had called. And he also these people, they also stated that these factors, they moved when the gametes were formed. Same was the case with the genes when they segregated, when chromosomes, homologous chromosomes, they separated. So this, a lot of further studies on genetics were done on Drosophila that you can see on the screen. These fruit fly or the Drosophila, why are they suitable? This question can be asked, like we had studied why is P chosen by Mendel. Same ways, why is Drosophila genetically suitable for studies? Because it can grow on simple synthetic media, it has a very short lifespan, the breeding can be done throughout the year, there is no season for breeding, hundreds of progenies are produced per mating, and male female can be easily distinguished since male Drosophila is smaller than the female, so it can be very clearly understood. The chromosomes that I was talking about, the chromosomes that you can see here, they carry genes. As you can see on the screen, this is a homologous chromosome, one from mother, one from father. Can you see the same location of gene? If the gene for eye color is at the upper position, so the same will be the position of the chromosomes here also, in the paternal chromosome also, right? So the position of the genes on the chromosomes is same in the homologous chromosome. Suppose this chromosome has gene for cytochrome C here, so this, the homologous partner will also have the same gene at the same location. That is why these chromosomes are called homologous chromosomes. And the genes, one gene is present here, another gene is present here, these are, we now know that we call them alleles, right? Okay, so now homologous pair that you see here, one from the mother, other one from the father, and when the cell division has to happen, this chromosome duplicates itself. इसमें DNA का amount double हो जाता है, so this chromosome duplicates at the centromere position. Now you can see this X-like structure in which these are chromatids. These are chromatids, right? Okay, and they carry the genes or the chromosomes which kept on moving. Okay, now coming on to linkage and recombination. Let me tell you one thing. This linkage is a term which is inversely proportional to recombination. Now what does that mean? Linkage का उल्टा होता है recombination. I always link your subjects with other subjects to make it interdisciplinary. जैसे मैंने आपको बताया था कि pressure is inversely proportional to volume. तो अगर volume बढ़ जाएगा तो pressure कम हो जाएगा, pressure बढ़ जाएगा तो volume कम हो जाएगा, right? So same ways here linkage is inversely proportional to recombination. अब linkage का क्या मतलब हुआ? अगर दो chromosomes के ऊपर अगर दो genes they are lying close to each other like this, if they are present close to each other, do you think crossing over will take place between them? Very rarely. So these genes are known as linked genes. Any two genes which are closely located on the chromosomes will be called linked genes, and they will show less recombination. They will have parental combinations, जैसे हमने F1 और F2 generation में देखा था, that we get new combination of genes. We had seen in law of independent assortment also in dihybrid cross कि अगर एक tall और एक yellow कलर की जीन है और एक dwarf और green कलर की जीन है parental combination में, so we see new combinations in case of F2 progeny कि हमको अब दो नए और पेरेंटल कॉम्बिनेशंस के अलावा दो और नए रिकॉम्बिनेंट्स मिलते हैं लाइक टॉल एंड ग्रीन, येलो एंड ड्वार्फ which were not there. तो ये रिकॉम्बिनेंट्स होते हैं एंड these recombinants are formed when there is crossing over happening. पर अगर दो जीन्स इतने पास पास हैं क्रोमोसोम में, do you think recombination will happen between them? Do you think crossing over इनके बीच में possible है? Very rarely. So these genes are now said to be linked genes. Now we can see this on the screen. On the screen, if I show you these two genes, I hope you're able to see this. These two genes, capital A and capital B, they are, you can see, very closely located. So these genes are linked genes. अब अगर यहाँ बात करें हम, if you see here, these A and B gene, they are quite far away from each other. So they are quite far away. So these are not linked. So they can have more combinations, right? They can form more new parental combinations. यहाँ पर भी same thing is shown here. As you can see, ये genes they are closely located. ये crossing over हो रहा है पर parental ही आ रहा है, ठीक है? यहाँ recombinants बन रहे हैं. All right, so here you can see if the crossing over is happening between the genes, then you see new recombinants. अगर ये gene ही बहुत पास पास हैं तो इनके बीच में recombination होना मुश्किल है. I hope now you would have understood the terms of recombination, understood the terms recombination and crossing over, linkage and recombination. और ये मॉर्गन ने एक्सपेरिमेंट किया था. दिस क्वेश्चन इज ऑफन आस्क्ड कि who worked on Drosophila? The scientist name is Morgan. Morgan Hunt. Okay, and then you can also be asked Morgan ने किस organism पे काम किया था? That's Drosophila or the fruit fly. So what did he do? इसने दो crosses करके दिखाए. I will show this. Now look at this diagram which is there in the NCERT, and it is quite confusing for students. So I want you all to listen to it very carefully and understand. So we have two crosses. We will here take two crosses. So first we will talk only about cross A. ठीक है? We will only be discussing about cross A. Cross A, you will see Y and W are two genes. ये दोनों genes बहुत पास पास हैं. As you can see, there's another white, yellow eye color and wild type, right? So this is a female and that's a male. तो जब इनमें cross होगा, so you will be able to see F1 generation में these Y and W, they are still together. Y plus and W plus, they are still together. तो यहाँ अगर हम देखें तो parental crosses, the parental cross percentage is very high, as you can see here, 98%. और जो recombinant बन रहे हैं, यहाँ आपको कलर से क्लियरली दिख रहा होगा, आधा स्काई ब्लू है, थोड़ा सा पार्ट स्काई ब्लू है, बाकी ऑरेंज है, राइट? and here you can see more sky blue and little orange. तो ये क्या है, recombinants बन गए. अब recombinants की percentage अगर हम देखें तो percentage of recombinants is very very less. What does that show now? This shows that these two genes are very much linked. Very much linked means they are physically located very close to each other on the chromosomes. That's why the crossing over between them is very limited, and these two genes are now said to be linked. Now I want you all to focus here on cross B. Now we see cross B, and we are considering here two other genes. One is for eye color, W, and the other one is for body size. I'm sorry, body size. And then you will be able to see that these two genes are quite away from each other, and you can see the parental in the F2 generation. In the F2 generation, you can see, since these two genes are quite far away, so they can easily have crossing over between them, right? So they can easily cross over. So we have parental combination 62% as compared to 98% here, and recombinants, the new parental combination, new combinations are 37% against this 1.3. So these two genes are far away, तो इनमें linkage भी कम है. So they are recombinant genes, or the genes showing less linkage. Less linkage means they are more prone to crossing over. Suppose this is one gene and that's the other one. Okay, so you can see cross happening between them. Cross happening, as a result of which the recombinants that you will be able to see will have this combination, right? So this chromosome is said to be recombinant, having two different regions, two different genes. So the genes which are linked will not show more recombination, and the genes which are far away will show more recombination. Now moving on to the next slide, you can see on the screen Alfred Stutwent. He was the student of Morgan, and Morgan had only was able to only describe linked genes. If the genes are quite close to each other and there is less recombination, so they are linked genes. Now what did this Alfred do? He did many crosses, and he was able to define the genes which are close to each other and the genes which are far away from each other. So by that he made genetic maps. If two genes showed little recombination, so he placed them together. If two genes are showing lot of recombination between them, he placed them apart. So by this method, he was able to map these genes on the chromosomes. So he was the first one to begin with physical mapping of genes on the chromosomes, which is even till now used in genetic maps. It is used in genome sequencing, where the whole genome of the individual is sequenced. Now coming on to the next topic, sex determination. The determination of gender of the progeny is called sex determination. We have three methods by which sex can be determined, or the gender of the individual can be determined. Number one is that in humans, XX and XY, we have been studying in since so many years that the females have the chromosome combinations XX and males have XY, right? So XX will form a female, right? Whereas XY will form a male, right? This will form a male. Okay, now talking about the other combinations. So we will be able to see birds. Birds will also have ZZ, ZW and grasshoppers. Moving ahead, sex determination in case of humans. So before we proceed, you know that humans have 23 pair of chromosomes. We have known this so many years ago. 23 chromosomes of chromosome pair in every human, right? Out of which 22 are autosomes. Autosomes means they do not carry any gene for gender determination. And one pair is allosome. As you can see here, one is an allosome or a sex chromosome, and the remaining all 22 pairs are autosomes. And this was given by Henking. Henking stated that the male 50% of the sperms receive this a nuclear structure which he called X bodies. Okay, so you can see, अब आप ये बता पा रहे हो ये जो chromosomes हैं, this chromosome, we call it homologous chromosome because one has come from mother, other one has come from father. This chromosome also, this homologous pair also, one from mother and the other one from the father. Correct? Okay. Moving on ahead, the sex determination in case of grasshopper will be XX and XO mechanism. XX, so female will be XX, as in case of humans also, it will have two X chromosomes, whereas the male will be only one X chromosome, so we call it XO. So we call it XO. So male is heterogametic as in humans also, and females are homogametic. So females will be homogametic. Correct? Moving on to the next one. Here XX and XO mechanism. Female will have two X chromosomes, as this condition is same in case of Drosophila also. XX will be female and XY will be a male. And in case of humans also, we have seen the same thing, 22 allosomes plus one sex chromosome. And in case of birds, the sex determination in birds is opposite to what we see in case of humans. Here males are homogametic. Humans में होता है ना, आपको पता है, XX will be a female. But in case of male, ZZ will be a male, but ZW, heterogametic condition, this will be a female. And ZZ will be a male. Alright. So we have understood these three type of sex determination, but there's another one very surprising thing is sex determination in case of honeybee. Do you know one thing? Honeybees, the male drones, we call male honeybee to be drone. Drone doesn't have a father, neither he has a son. Do you know this? Okay, let's explore this. तो फीमेल हनी अगर एक हनी बी को देखेंगे, so female will have 32 pair of 32 chromosomes, that is 16 pair of chromosomes. This is there in case of a female. But male को देखेंगे अगर हम, so male will have only 16 chromosomes. इसका मतलब female will be diploid, male is haploid. So such type of sex determination is called haplodiploid sex determination. Now what does that mean? इसका मतलब हो गया कि अगर एक फर्टिलाइजेशन से अगर कोई एक इंडिविजुअल बन रहा है, कोई एक हनी बी बन रही है, so that will always be a female honey bee. Now you see a queen, a female honey bee, and a drone, male honey bee. So when they fertilize, so fertilization will lead to development of female only. So after fertilization only female will be developed, which can be either worker bee or queen bee, depending on what food is she fed on. If that female is fed on royal honey, so this will become a queen. If that's fed on a normal wax, normal honey, so it will become worker bee. But agar fertilization ke bina hi, if the female has given eggs, they ultimately develop into drones, unfertilized eggs. So they are unfertilized. So that means a drone did not have a father because it developed directly from the female by what? Mitosis, right? By the process of only unfertilized egg being converted into a drone. Now if we move ahead, when this drone sometime will get fertilized with this female, so this will again form on fertilization again female only, worker bee, female worker bee or female queen bee, but never a drone. So it means never a drone. So this shows that a drone did not have a father, did not have a father and will not have a son. Rather, he will have grandchildren and grandfather. I hope you could understand this. Now, moving ahead, these are some of the competency based questions that I want you all to look at it and please answer yourself. For more clarity, we will have a online session, online live session as and when you people send me a comment message. Thank you so much. Please like and subscribe the channel. Thank you.